Welcome to the Deep Dive. We're here to unpack complex information, find those key insights, and really give you that shortcut to being well-informed.
And today, we're going deep, right into our own cells.
Yeah, specifically looking at how they build proteins. You know, the absolute workhorses of life. It's incredibly complex stuff.
It really is. When you think about it, billions of these precise operations happening every single second inside us. Proteins do almost everything, structure, signals. Building them is this incredibly coordinated dance.
It is. And think about this. What if you could see, like right now, which specific little molecular workers, these things called tRNAs.
The delivery trucks, basically.
Exactly. The delivery trucks bringing the amino acid building blocks to the protein factory. What if you could see which ones are actually working at this very moment? Not just, the whole fleet of trucks parked in the depot, but the ones out making deliveries.
Well, for the longest time, seeing those specific ones, the ribo-embedded tRNAs, the ones literally inside the ribosome factory while it's working, it seemed almost impossible.
Like trying to see which wrench a mechanic is using while the engine's running.
Precisely. Not just glancing at the toolbox. The whole process is so dynamic. These fleeting interactions, isolating that specific moment was a huge challenge.
Right. But that's what makes today's deep dive so interesting. Because that hurdle, that almost impossible challenge, well, there's now this cutting-edge way to tackle it.
It's called Ribo-tRNAseq.
Ribo-tRNAseq. And it's really a unique workflow, isn't it? It seems to be changing the game.
It really is. So our mission today is to unpack what Ribo-tRNAseq actually is, how it manages to do what it does, and crucially, why it's such a big step forward for understanding health and disease.
Okay, let's get into it. So this Ribo-tRNAseq, it's not just one thing, right? It's kind of a combination.
Exactly. It cleverly leverages two key technologies working together. Think of it as a really effective tag team.
Okay.
First up, you've got something called RebelSTM. Now, this is an active ribosome pull-down technique.
Cool down, so it's like grabbing them.
Yes, essentially. It lets researchers physically grab hold of the ribosomes, those protein factories, but only the ones that are currently active, currently building proteins, along with everything attached to them.
Got it. So you're not grabbing idle factories, just the busy ones.
Precisely, and that's crucial.
Yeah.
Then the second part of the team comes in, nano-tRNAseq.
Okay.
This is a very advanced tRNA sequencing technology. Its job is to then read or identify the specific tRNAs that were attached to those active ribosomes you just pulled down.
Ah, I see. So Ribo-tRNAseq grabs the active factory and its workers, and nano-tRNAseq identifies exactly which workers were on the job.
You've got it. And that combination is what gives us this unprecedented view.
And you mentioned it's efficient. How quick are we talking?
Remarkably quick for this level of detail. It allows researchers to go from their starting material, the cell, basically the cell contents.
I wrote the soup.
Yeah, the soup, to getting comprehensive data back in just a few days. The kit they offer quotes a 3-day workflow time.
3 days. That's really moving for molecular biology of this complexity.
It is. It makes it much more practical.
So for labs thinking about this, what do they need? Massive samples, super specialized gear.
Actually, it's pretty accessible. For cell lines, you need upwards of about 500,000 cells, which isn't huge. For tissues, maybe more than 10 milligrams.
Okay, that sounds manageable.
Yeah, and the kit comes with things like the annotated genome information and the necessary license, which helps streamline the whole analysis part afterwards. The catalog number, for those interested, is RLNTR12, and it gives you 12 reactions.
Good practical details. Okay, so we know the key players, Ribo-tRNAseq and nano-tRNAseq. Can we walk through the workflow, step by step, from cells to data?
Sure. So step 1 is preparing your sample cell or tissue lysis. You gently break the cells open.
Release the contents.
Yep. Then comes that key step, the Ribo-tRNAseq active ribosome pull down. That selectively grabs the active ribosomes and their bound tRNAs.
Isolating the important bits.
Exactly. After that, you need to clean things up. So RNA extraction to get the genetic material out, followed by small RNA enrichment.
Why small RNA enrichment?
Because tRNAs are quite small compared to other RNAs, like messenger RNA. This step helps concentrate the tRNAs and get rid of, well, bigger stuff you don't need for this analysis.
Makes sense.
Yeah.
Focus on the target. What's next?
Then the nano-tRNAseq part really kicks in. This is the library prep stage.
Library prep, like preparing the RNA to be read by the sequencer.
Precisely. It involves a few steps. First, retrotranscription, turning the RNA back into a DNA copy, which is generally more stable and easier for sequencers to handle.
Okay.
Then you add special molecular tags. They call them splin adapters and barcoded adapters.
Barcoded. like for scanning at a supermarket.
Sort of. The barcodes are unique molecular tags. They let you mix several different samples together, up to 6. With this kit, run them all in one sequencing reaction.
Ah, multiplexing. Saves time and money.
Exactly. And then the sequencer can read the barcode and know which sequence came from which original sample.
Clever. So library prep is done, samples are barcoded, then sequencing.
Then sequencing, yes. Typically on an Oxford Nanopore platform for this specific method.
And once the raw sequence data comes off the machine,
It's data analysis time. You use a demultiplexing tool first. That's software that reads the barcodes and sorts the data back into the original samples.
unscrambles the mixed batch.
Right. And then you use specialized analysis tools, often provided or recommended with the kit, to interpret all that sequence information and figure out the abundance and types of those active tRNAs.
Okay, that gives us a really clear picture of the how. Now let's shift to the why. Why is getting this specific information, the active tRNAs, so important? What doors does Ribo-tRNAseq open?
Well, it lets us ask some fundamentally new questions. We can finally, reliably measure the abundance of active tRNAs. And not just abundance, but also their modifications, which can be really important for their function.
Modifications,
Yeah.
like little chemical tweaks to the tRNA molecules themselves.
Exactly. And those can change how they work. So we can see which modified tRNAs are being used. This also lets us explore how these actively used tRNAs might play a role in various diseases.
Okay, that's huge.
And we can track dynamic changes. How does the cell shift which tRNAs it's using when conditions change, during different cell states, or when it's stressed?
So watching the cell adapt in real time at the level of protein synthesis?
Pretty much. Plus, as we mentioned, the practical side, it's a gel-free, fast, multiplexing workflow. That makes these kinds of studies much more feasible for labs.
The real test is always the data, though. You mentioned some case studies. Do they back up these claims of sensitivity and accuracy?
They do. Let's look at the first one, which focused on reproducibility and specificity. They used HEK293 T cells, a common cell line in labs.
Okay.
The reproducibility between replicate experiments was incredibly high, an R-squared value of 0.98.
Wow, 0.98 is near perfect correlation, so it's reliable.
Very reliable. But maybe even more importantly, it showed specificity. It could accurately distinguish the tRNAs that were actively involved in translation from the general pool of all tRNAs just sitting in the cell.
How did they show that distinction?
They used a statistical method called principal component analysis, or PCA. It's a way to visualize complex data. And the PCA plot showed 2 very distinct clusters.
Clusters.
Yeah, groups of data points. One cluster, shown in orange in their figure, represented the ribo-tRNAs, the ones pulled down by ribolus. The other cluster, in blue, was the total tRNA from the cell. And these 2 clusters were clearly separated.
Meaning the technique really is isolating a specific population, the active ones.
Exactly. It's not just measuring everything, it's successfully targeting the tRNAs caught in the act of translation. That's a critical validation. It gives us confidence that we're seeing something biologically meaningful.
Okay, that's specificity covered. What about detecting changes? Can it see things shift?
That's what the second case study looked at. They used a different cell line, MCF7 cells, and treated them with a drug called Harringtonine, or HAR.
Harringtonine.
What does that do?
It's a known translational inhibitor. Specifically, it stops the ribosome right after it starts building a protein chain. It arrests C-elongation just after initiation.
Okay, so it jams the factory near the beginning of the assembly line.
A good analogy. Now, when they looked at the total tRNA levels in the cells before and after HER treatment, things looked pretty much the same. No big changes.
So the overall supply of tRNA trucks was stable.
Right. But when they used Ribo-tRNAseq to look specifically at the tRNAs on the ribosomes,
Yeah.
they saw significant changes, particularly in the levels of initiator methionine tRNA, or IMET-tRNA.
A tRNA that starts protein synthesis.
Exactly. The levels of active ribosome-bound IMET-tRNA changed significantly. And this makes perfect sense because HER stops things right after initiation, so you'd expect a pileup or change related to that starting tRNA. So Ribo-tRNAseq picked up a dynamic shift that was completely invisible if you just looked at the total tRNA pool.
Precisely. It demonstrates the method's sensitivity to detect these important functional changes in real-time cellular processes. It shows the cell reacting to the inhibitor exactly where the inhibitor acts.
That's compelling evidence. What about a more sort of natural stress, not just a drug?
Case study 3 tackled that. They used those HEK293 T cells again, but this time they subjected them to nutrient stress. Specifically, they deprive the cells of either leucine or arginine for 3 hours.
Two important amino acids. Leucine affects translation initiation. Arginine affects elongation. So starving the cells of these should definitely impact protein building.
Absolutely. And again, they compared these starved cells to control cells that had all their nutrients.
And what did Ribo-tRNAseq show?
Once more, the PCA analysis was key. It showed clear separation between the different conditions, the fully fed cells, the leucine-starved cells, and the arginine-starved cells. Their active tRNA profiles were distinct.
So the cell changes which tRNAs are active on the ribosomes, depending on the specific nutrient it's missing.
That's what the data strongly suggests. The method is sensitive enough to pick up these specific adaptive responses. And they could even pinpoint changes in specific tRNAs like GLIA-CCC, LuCAA, and that initiator IMEPEC again.
Wow. Seeing those specific tRNA changes under different nutrient stresses, that's a level of detail about cellular adaptation that was, well, pretty much inaccessible before, right?
Completely inaccessible at the scale and specificity. You might have inferred things indirectly, but actually seeing the shift in actively used tRNAs like this, it's new.
So let's recap. We've gone from a situation where studying these ribo-embedded tRNAs was deemed nearly impossible.
A black box, really.
To now having Ribo-tRNAseq, a tool that lets researchers put this analysis right on their bench. It seems like a massive leap.
It absolutely is. It's opened up this whole area. Understanding the abundance, the modifications, the dynamics of the tRNAs actually doing the work. It gives us a much clearer view of how translation is regulated.
And potentially, how it goes wrong in disease.
That's the crucial link. Dysregulation of protein synthesis is implicated in so many diseases. This tool gives us a way to probe that directly.
Which brings us to that final provocative thought for you, our listeners, to consider. Now that we can precisely measure which tRNAs are busy building proteins.
Which delivery trucks are on which routes in real time.
Right. What new insights could this give us into really complex diseases? Think about cancer or neurodegenerative disorders like Alzheimer's, where we know protein synthesis can go haywire.
Could understanding these dynamic tRNA shifts lead to new biomarkers or even entirely new therapeutic strategies aimed at correcting translational problems?
How might this detailed view reshape how we target those diseases? The possibilities seem, well, truly vast. Something to ponder.